Hi, everyone. My name is Luiza. I'm a PhD candidate with Sebastian Huna at the LMU in Munich, Germany, and I'll talk about convergence assessment in Bayesian phylogenetics. So if you attended the workshop this morning, you already saw some of these concepts. This is Bayes' theorem, which states that the posterior probability of the parameters equals the likelihood of the data times the prior probability of the parameters divided by the marginal likelihood of the data. And in the case of phylogenetic inference, the data is sequence alignment, and the parameters that we are estimating are the model parameters, such as the stationary base frequencies, the tree length, the clock rates, and of course the phylogenetic tree. But there's a problem, and that is this marginal likelihood is a multidimensional integral, and we cannot solve this analytically. And to overcome that, we use Markov chain Monte Carlo algorithms. So we draw samples from a probability distribution based on a Markov chain that has a desired distribution, a stationary distribution. And by Markov chain, we mean that the current state depends only in the exact previous state. So as a very simple toy example, here on the x-axis, we have the parameter space, and this distribution represents the true distribution of a parameter. And we want to estimate the posterior probability distribution of this parameter. So we start at a random point in the parameter space, and the MCMC set the rules if we can move to another point of the parameter space or if we should stay in the same place. And we move to another point, and we ask the same question to the MCMC again. And in that way, we move around until we have covered the parameter space, and the amount of iterations that we spend at a given point is gonna be proportional to the posterior distribution. And that imposes that we need to check for convergence after running an MCMC, because we need to make sure that we took samples from the stationary distribution and that we have enough samples for estimating the parameters reliably. And by convergence, we mean convergence in distribution. And by the example in the figure, we can see that the more samples we take, we have a better understanding of how the distribution looks like. But there's also no mathematical solution to calculate how long the MCMC should run. Instead, we check for convergence by examining the output from the analysis. And a common way to do that is calculating the effective sample size, which is the number of independent samples. So the number of samples that are not correlated. In practice, this is done by dividing the number of samples by the autocorrelation time. We also use plots of the states of the parameters across MCMC iterations. So this is a screenshot of Tracer that some of you have probably already used. And here's an example of the states of the tree length across the MCMC iteration. The potential scale reduction factor is a test from the statistical literature, which compares the variance within and between chains, and the values should be close to 1. So here, as we see in the figure, as we increase the number of samples, the PSRF approaches 1. And the average standard deviation of split frequencies is specific for phylogenetics. And a split can be defined as a partition of the tips of the tree into 2 non-overlapping sets. So for example, for this blue tree, we have the splits AB, ABC, and the full tree, ABCD. So we calculate the average standard deviation and we set up a threshold for that value. And what are the problems with the current methods? First of all, the statistical diagnostics have been shown to fail even for simple problems. And in the figure that I showed before, we can see that depending on the distribution, the PSRF varies a lot. There's a lack of clear thresholds for the PSRF, for example, also for the ESS and the average standard deviation of split frequencies. There's no straightforward interpretation of the plots. We also have the problem that we need to manually check the analysis. So when we have many analyses, we have to upload them individually to the tools and check. It requires visual inspection. And for the average deviation of split frequencies, the average can fail to capture extreme situations. And all that culminates into poor description of convergence status, which leads to no reproducibility of Bayesian phylogenetic inference. So we propose a new approach based on two characteristics that we want to have in our MCMC output, and those are precision and reproducibility. And by precision, we mean that more samples do not change our estimates. And with reproducibility, we mean that another MCMC replicate lead to the same estimates. And how do we check for precision? We calculate the ESS. So how many independent samples do we need for a precise estimate? So we base that on the standard error of the mean, which is a measure of precision of the mean estimate. And if we consider a normal distribution, the 95% interval corresponds to 4 times sigma. And if we establish that the standard error of the mean has to be smaller than 1% of the 95% interval, we end up with an ESS of at least 625. So what that represents, if we establish the width of the standard error of the mean to be 0.5%, that is represented in the figure on the right. That's how much we are allowing our mean estimate to vary. We would need an ESS of 2,500. For 1% and an ESS of 625, that's how much we are allowing the standard error of the mean to vary. And as we increase the width and decrease the ESS, this interval gets larger. In case of the trees, we check the ESS for the splits. So we will check the presence or absence of a given split in the sample trees. So we have our MCMC iterations, and we sample this red tree, and we check that we have the split AB and the split CD. So we attribute a value of 1 for the splits that we have and 0 for the splits that are not present. And as we sample trees, we do the same for the splits. And we end up with a vector of 1s and 0s, which is similar to a binomial probability. And then we can calculate the ESS with the standard methods. But there are different methods to calculate the ESS implemented in different tools. For example, for CODA, MCMC, SE, and Tracer, they implement differently the way they calculate the autocorrelation time. So for CODA, they calculate the autoregressive process and the spectral density at frequency 0. For MCMC, SE, they use a batch min approach, and tracer uses an initial sequence estimator. So we had to decide which method to use, and we decided to test for independent samples. So drawing independent samples for different distributions and then checking the estimated ESS with these different methods. And we see that tracer tends to underestimate the ESS, and CODA estimated with more precision in the case of independent samples. We also tested for the binomial distribution, since that's what we are using for the splits. So we tested for different probability of success P, and they all produce the same profile. Again, tracer is underestimating the ESS. But in the MCMC, the rule is that we have autocorrelated samples. So the samples are not independent. So we decided also to test the estimation of ESS in case of autocorrelated samples. So we developed an algorithm to create autocorrelated samples, and we did the same as before. So we draw an initial independent sample, which corresponds to the true ESS, and then we added correlation to that sample and estimated the ESS. And here we can see the tracer was less affected by autocorrelated samples. And this is for the normal distribution. We also tested for the binomial distribution, but here this figure represents a different test. So we fixed the initial sample size for 625, and we varied the autocorrelation and the probability of success P. And also for the binomial distribution, we see the tracer was more stable and reliable to estimate the ESS. And I mentioned that we would look into two aspects of the MCMC, precision and reproducibility. So to check the reproducibility of the continuous parameters, we implemented the Kolmogorov-Smirnov test, which is a non-parametric test, and that means that it's independent of the distribution. And for an alpha of 0.01 and an ESS of 625, which is the default value that we established for the precision, the KS threshold is 0.092. So for example, if we have these two figures here representing two distributions, I think most of us would agree that the left one, the samples don't come from the same distribution, but on the right one, we would probably not all agree on the answer. So applying the KS test, both would have failed, so both samples don't come from the same distribution. But for the splits, we have discrete parameters, so we cannot apply the same test. Instead, we calculated the expected difference between splits, and that is based on the mean absolute difference between two binomial distributions. So we calculated this expected difference based on different true probabilities and with different ESS values. And we can see first that the expected difference is going to depend on the true split frequency. And also that as we increase the number of samples, the expected difference is going down. And all of that is implemented in an R package called Convergence. It accepts MCMC output from RevBase, MrBase, Beast, and Phylobase currently. And the results provide a complete summary of the convergence assessment, as well as tables and also plots. And this method that I described here is all implemented in one wrap-up function called check convergence. So you just need to provide the path to the files or the list of files, as well as the format of the files. And the function gets the precision. So the default value is 1%. And that means an ESS of 625. And then the function calculate all the thresholds for the tests, loads the output from the MCMC, checks if the burning is appropriate, and then it computes the ESS for the continuous parameters and for the splits, the KS scores in case of the continuous parameters, and the difference in split frequencies. And then it finally reports if convergence was achieved or not. So the nice feature is that we can have the results of the convergence assessment in plots. For example, here is the histogram of the ESS for the continuous parameters and for the ESS. So the dashed line represents the threshold, and we expect all values to be above the threshold. So this is very easy to report that convergence was achieved. We also plot the histogram for the KS test. And again, the dashed line is the threshold, but here we want all values to be below the threshold. And for the difference in splits, we plot the difference between splits against the split frequency. And the gray line represents the threshold for the ESS of 625. And we expect all splits to be below this threshold. So the package is open source, is on the GitHub page. There's also under the page of RevBase a tutorial. And with that, I would like to thank you for the attention and open to questions.